The goal of this experiment is to acquire two-dimensional instantaneous fields of velocity in Hagen-Poiseuille flow, also known as laminar pipe flow, using echo particle image velocimetry, or EPIV. EPIV validation measurements are demonstrated in a recirculating pipe flow of a 50-50 mixture of water and glycerin. The pipe flow system used here employs a constant pressure head maintained by an aquarium pump to drive the flow. A phased array ultrasound probe is mounted to the pipe wall of the flow system, and B-mode ultrasound images are streamed. The fluid is seeded with hollow glass spheres or tracer particles that faithfully follow the motions of the flow. Ultrasound images are then acquired, transferred to a PC, and converted to an image format compatible with the commercial particle image velocimetry software. Cross-correlation algorithms are applied to successive ultrasound B-mode images to compute two-dimensional fields of velocity. Ultimately, the vector fields are analyzed to compute flow quantities of interest, such as ensemble average, shear stress, and vorticity.
Though this technique can provide insight into basic fluid dynamics, it can be and is often used in practical flow systems, including biomedical applications, for example, arterial or intraventricular flows.
Our ongoing experiments using liquefied biomass and opaque fluid is what interested us in this technique.